So we're going to start with thinking about what is hypersensitivity. So this is an inappropriate reaction, meaning that the immune system is usually just doing its job. It's trying to protect us from something toxic, something foreign, or trying to initiate repair for damaged tissue. But if that response is out of proportion to the stimulus, then it's considered inappropriate. Oops. And then second is if that response causes damage to the body beyond what is needed for eliminating a pathogen, meaning it causes injury, then we term it hypersensitivity. So hypersensitivity reaction is really an excessive reaction of the immune system. And this is a picture here. Does anybody recognize that? What might that be? Hives, yeah. So if you've had it, you probably recognize this. And you'll notice this doesn't look much different from other types of inflammation. So you have swelling in discrete areas. These are called wheals clinically. You have redness all around this area. And if you were to put your hand on it, you would feel that there's warmth in that area. So all of the signs of inflammation are there. It's also extremely itchy. That itchiness is similar to pain. It's caused by the release of cytokines, especially histamine. So hives are a type of hypersensitivity. Anybody here have hives? Several people. What do you react to? What do you react to? Penicillin. Do most of us have a response to penicillin like that? No. So your response is hypersensitive. It's hyperactive immunity to what should be a non-toxic stimulus. What else do people respond to? Yeah? Cats. Right. Most of us can handle a cat and not break out in hives or get these crazy, watery, itchy, swollen eyes. But some people are allergic to cats. And that's because your immune system has an exaggerated, an inappropriate reaction to that stimulus. So when we're thinking about what would trigger a hypersensitivity reaction, there's 3 classes of antigens. Remember that the immune system is responding to antigens. It's responding to something in the body. Now, most of the time, we've already learned, the immune system should not respond to self-antigens. However, if something goes wrong with that maturation process, where T cells and B cells are tested against self-antigens, if something goes wrong there, then you can develop an immune response to self-antigens. And when that happens, we call that autoimmune disease. So this is really a failure of self-tolerance. Now, sometimes you have an exposure to microbes, and you fight that microbe, and it goes away. That's a normal immune response. But some people will have a hyper response. So the microbial antigen is appropriate, but the immune response to it is inappropriate. So an example of this is if you are ever diagnosed with strep throat, your doctor will recommend you take antibiotics. The reason is not because your body can't fight strep throat. Your body can fight Streptococcus pyogenes, which is the bacteria that causes strep throat. But what happens is as you're fighting that bacteria, you produce antibodies to those microbial antigens. And in some people, sometimes those antibodies cross-react and start to recognize other tissues in the body, such as the heart. So rheumatic fever is a hypersensitivity reaction to a normal microbial antigen. So you have really an exaggerated. An exaggerated response to something that you would want your immune system to normally attack. And then the third class are environmental antigens. And these are what we just heard people with hives tend to respond to. So environmental antigens are things that don't cause disease in most people, but for some reason, in some people, they trigger an immune response. So cat dander is a good example. Peanuts, peanut allergy. Most of us can eat peanuts with no problems, but in some people, peanuts can cause a very severe allergic response. So this is a hypersensitivity to a otherwise harmless antigen. Now, be really careful with environmental antigens, because if it's something that causes damage to everybody, we would call it a toxin. So environmental antigens are not toxins. For example, if any of us get bitten by a rattlesnake, we can expect to have a pretty bad response to that. That's because rattlesnake poison is toxin. It's toxic to the body. So that's not an environmental antigen for hypersensitivity. That response that any of us have is a toxic response. It's not a hypersensitivity response. Yes?
What about like a stink from a bee or a wasp? Like, not everyone reacts to it, like some people
Right.
have more of a...
So good question. So a bee sting is a perfect example of an environmental antigen, because when a bee stings you, you get a little bit of tissue damage. And most of us would have an appropriate immune response, which is a little bit of redness and swelling in the area. That inflammation is coming in to eliminate the damage and the environmental antigen that's in the area. That's normal. Some of us will have an abnormal hypersensitive response to that, and it can trigger anaphylaxis. So that's a hypersensitivity. The normal little bit of redness there, it is an appropriate reaction. Yes?
Your body change what it's allergic to?
Yes, your bodies do change what you're allergic to. And I was trying to do some reading about that because somebody else asked. And I think it's still kind of a mystery. So why do some people, they're fine with cats until they're about 25 years old, and all of a sudden they develop allergies? Or for anybody who's ever worked in a mouse or a rat lab, I used to do research with rats. 90% of people who do research with rats or mice become allergic to them after working with them for several years. But we don't exactly know why that is. It's a great question. Maybe you can do research to figure it out. Yes? Can you get over an allergy? I think the answer is yes. Has anybody here been allergic to something as a child and now they're not? Okay, so empirically, yes, you can get over an allergy. And there's a lot of ongoing research into desensitizing people to allergens. So I know that they give like injections to get over a cat allergy or a peanut allergy, where they expose your body to small, tiny amounts of the antigen, and then they increase the amount of that antigen that you're exposed to in order to teach the body not to overreact, not to respond. Whether you can do that without, medical intervention, if you could just do that yourself, is still an ongoing question, but there are reports out there, and I linked to an article from the New Yorker that kind of talks about this a little bit. In infants with milk allergy, they're now recommending you cook food with a little bit of milk in it and try to get the child exposed to small doses, and then you can increase it over time and try to at least reduce the allergy response to that protein. So it can change over time. but the mechanisms through which that occur are still kind of being worked out. But it is really interesting because allergies are on the rise. More people today have allergies than who lived 100 years ago, and especially more than people who lived 1,000 years ago. So why is that happening? People are looking into those mechanisms. But just a little bit about peanut allergy along those lines. How many people have heard that you should not give infants peanuts until they're 1 year old? So that was, when I had my own kids, that was the recommendation. The American Pediatric Association in 2000 posted that as a recommendation for all parents not to expose their children to eggs, milk, peanut, a bunch of other things until after they were 1 year old, thinking that the immature immune system can't handle it and is more likely to develop an allergy to it. When my twins were born, my third and fourth child, my doctor said, there's some new research out showing that people in Israel have much lower rates of peanut allergy than people in the UK or people in the United States. In Israel, they eat peanut butter as one of their very first foods. So young children, 4, 5, 6 months old, start eating peanut types of snacks. And the rate of peanut allergy there is incredibly reduced compared to other places. Yes? Honey is a different thing. So the peanut thing turns out that you actually want early exposure in increasing doses. The problem with honey is that if it's raw, it's more likely to have botulism. And a baby will have a very severe response to that because their immune system is reduced. Whereas once they're a year old and their immune system is more mature, if they're exposed to small amounts of botulism, they can handle it. And all of us can handle small exposures. Yeah, question over here? Richard?
For the, back to the peanut butter one, could it be, not to be morbid, but could it be that the ones that died from eating peanut butter didn't grow up, and so now they're not immune to it?
That's a great hypothesis. Is it that now we're saving more babies that should not have, or should is the wrong word, but that would not have survived previously? That probably does contribute to it, but it wouldn't explain the difference between Israel and the UK or the United States, because we save the same number of babies in both countries. I just heard on NPR a few months ago that they've now changed the recommendation, and now they do encourage you to start giving your babies peanut products early on and before they reach 1 year old. So these recommendations are constantly in flex. I think what it really illustrates is that we don't understand the immune system very well yet. Okay, so let's talk about these types of hypersensitivity reactions. The type of reaction is divided based on the mechanism underlying it. Clinically, there's a lot of overlap. Also, you can have more than one hypersensitivity type going on at the same time. So really, we're dividing them by mechanism right here. So immediate hypersensitivity is what we've just been talking about. These are your allergies. We'll talk about the mechanism in just a few minutes. Also included in this is asthma and atopic dermatitis. These three together are your atopic triad. So this is what is on the rise in industrialized nations, are these diseases. And it's, if you have one, you're more likely to have another. So if you have atopic dermatitis, you're more likely to also have asthma, or you're more likely to have a family member with them. So there is some kind of genetic predisposition to this. Yeah?
About like weather allergies, like I'm
Yes, so there are other types of allergies, and she was asking about weather-related allergies. So the most well-studied is cold-induced asthma. So some people will only develop asthma if they're trying to exercise in the cold, for example, or with changing seasons. The mechanism underlying there is a little bit different than what we're talking about here. And I don't know that it's completely understood why some types of stimuli that are not antigens, or if there's some other underlying antigens in the weather change that is triggering those types of reactions. But there are cells of the body that can respond to stimuli such as cold differently than most people's cells, and somehow that triggers an immune response in people with cold-induced asthma, for example. But I don't exactly know the answer to why some people just get more of the sniffles if it's not a hay fever type reaction. So that's immediate hypersensitivity. It's mostly mediated by IgE on mast cells. We'll talk about that in just a minute. The second type, type 2, is antibody-mediated hypersensitivity. And this is where antibodies floating through the body will bind to your own tissues. They bind to your tissues, and they trigger a response. Again, that's what they're supposed to do, right? They're supposed to bind to antigens and trigger a response. But if those antigens are in the wrong place, or if they're self-antigens, then you get hypersensitivity. So antibody-mediated is where the antibodies bind to tissue and trigger their normal inflammation. Now, that's different from immune complex mediated, where the antibodies bind to their antigens in the blood. So remember, antibodies are constantly circulating in the blood after they've been formed. They will bind to antigen that's also circulating in the blood. So that's called an immune complex. These immune complexes can get deposited in various tissues. So these are antibody-antigen complexes deposited in tissue. Otherwise, antibody-mediated and immune complex-mediated are very similar. So it's really just depends on where the antigen is in those two types of hypersensitivities. And then the fourth type is T-cell mediated hypersensitivity. So we already learned about the two different types of T cells. So you can have T helper cells that secrete a lot of cytokines and induce inflammation. Or you can have cytotoxic T lymphocytes, which are your CD8 positive T cells that trigger apoptosis. So the first three types are really antibody-mediated, and then the last type are all of the T-cell mediated reactions. So what types of hypersensitivity reaction are going on in transplant rejection? We don't want to reject this organ, so it's inappropriate and injurious, so it's hypersensitivity reaction. What type? Yes. Yes. So type 2, which is your antibody-mediated. Because remember we said with transplant rejection, you develop antibodies, mostly, what's the protein target or the antigen target for those? Usually? In the donor tissue? Yeah, so usually it's MHC. So these MHC proteins on the surface of all donor cells are immunogenic, and they trigger an antibody-mediated inflammation of the organ. Any other types you think might be involved?
We didn't think type 3 would be because you're not getting the going blood, you're just getting the tissue. So
Right.
Nothing floating around, so it would be 4.
So usually it's not going to be immune complex mediated. That requires the antigen to be floating around. It can be, but that's not usually what's going on. What about other types, type 1 or 4?
Wouldn't type 1 have something to do with it because you said it can reject it within seconds to minutes?
So the hyperacute, that's a great question. Would hyperacute, because it's immediate, like type 1 hypersensitivity. The difference is with hyperacute rejection, these are preformed antibodies. Usually they're IgG or IgM class antibodies that are already circulating in the recipient's blood. With an immediate type 1 hypersensitivity reaction, these are IgE antibodies that are priming mast cells. So when we get into the pathogenesis, I think you'll see a little bit more, but it is a little bit different. So a hyperacute rejection is still the type 2 antibody-mediated rejection. It just happens so fast because those antibodies are already present in the recipient's blood, rather than needing 3 to 5 days for B cells to generate them. So type 1 is actually not involved in tissue rejection or transplant rejection. What about type 4? I see some people nodding their head. How many people think type 4 is involved? Yeah? Okay, you're right. So type 4, which is T cell mediated, and again, these T cells are primed and recognizing MHC as well. You'll find that type 2 and type 4 often go together because, as you know, helper T cells help to activate B cells. So it's not unusual to see T cell mediated and antibody mediated responses going on at the same time. How about autoimmune? Which types are involved in autoimmune? Which one? Type 4? Yes. Many autoimmune diseases are T cell mediated. So anytime the T cells are recognizing self-protein, and these can be helper T cells or cytotoxic T cells, you will get this type 4 hypersensitivity that is classified as an autoimmune disease because it's a self-antigen that's activating them. Any other types could be involved? Yeah. Type 2. Again, often you'll see type 4 and type 2 going on together because they kind of help activate each other. So in autoimmune diseases, you'll frequently see autoantibodies, antibodies directed against self-antigens. And if those antibodies are directed against antigen in a certain type of tissue, they'll bind to the tissue, trigger inflammation in that tissue. What about type 3?
Why not type 3? You can't think of why not. So.
Why not? Yeah, type 3 can also be involved. So type 3, if you produce antibodies that recognize an antigen that is part of like proteins that are just circulating in the blood, then you can develop antigen-antibody complexes in the blood, and those complexes can deposit. And we're going to see that with lupus because that happens a lot in lupus. The only one that you don't usually see in autoimmune disease is type 1. So type 1 is really your allergies. It's really responding to an external stimulus in an inappropriate manner. Autoimmune disease is a response to any self-protein. So any of the other types of hypersensitivity can be contributing to autoimmune disease. Okay, let's go through each one in a little bit more detail now. I think I had a question for you, but I want to look at it. Okay. So before we go on, think about these 4 different types of hypersensitivity reactions that I just mentioned. Which one or which ones do you think are important in transplant rejection? And which one and which one or which ones do you think are important for autoimmune diseases? So I want you to take 2 minutes, talk to your neighbor, and try to figure out which types of hypersensitivity are most likely mediating transplant rejection, which we talked about last Tuesday, and which types are involved in autoimmune diseases. So let's look at immediate hypersensitivity. So I just told you the primary trigger for immediate hypersensitivity is environmental antigens. So these are things that we are exposed to in our everyday life that in the majority of people do not cause any harm. But in some people, for some reason, they trigger an immune reaction. And the cells that are triggered in this immune reaction, it requires a coordinated effort. So initially, you have T helper cells. These T helper cells will activate B cells. The B cells are going to secrete IgE, which is going to bind to mast cells, as well as eosinophils. I'm going to abbreviate as EOs. So lots of cells are involved in type 1 hypersensitivity. So this is taken from your book, and it's a really good drawing to think about the pathogenesis of what's going on. And I'm going to get into the pathogenesis in just a second. But notice here, so it's IgE antibody mediated, so it has antibodies. These IgEs are recognizing this environmental antigen. And what happens is, after they're produced, they bind, mast cells and eosinophils have receptors on their surface that recognize the Fc portion, that constant region of IgEs. And remember, the constant region is antibody type, antibody class specific. So IgGs, IgMs, IgAs, they won't bind to mast cells, only IgEs. But it can be any IgE. That's why some people are allergic to cats, and some people are allergic to mold, and some people are allergic to peanuts, because they're all producing different IgEs to that specific environmental antigen. Once those mast cells have IgE on them, if they're exposed to anything that binds to that IgE, so that specific allergen, you're going to cluster them, and that causes those mast cells to immediately degranulate. So all of these little dots in here are granules that are filled with things like histamine. And as soon as it's triggered, it's going to dump histamine. So if you think back to what I showed you with that hives picture at the beginning, what that was is every single one of those wheels is where a mast cell or a small group of mast cells were triggered inside the body, underneath the skin. So those mast cells, they had IgE already on them. The allergen got into the body and caused those mast cells to immediately release those granules. And histamine is a very active, it's called a vasoactive amine because it causes vasodilation and increased vascular permeability. And this shows a little bit more detail about what's going on with that process. But what's really important for type 1 hypersensitivity is that it requires priming. So most of the time, if you haven't been exposed to an allergen, you do not have IgEs for that allergen. So how many of you have heard that if you're allergic to strawberries, for example, the first time you eat strawberries, you don't actually have a reaction. It's the second time. So you don't have a reaction the first time you're exposed to an allergen. The reason is because that allergen has to first stimulate the production of IgEs. The allergic response is in response to those IgEs. So let's say you have your first exposure. Your antigen comes into the body, and it might get recognized by a helper T cell. That helper T cell is going to secrete cytokines. Now, here's a good question, and I actually don't know the answer. I don't know if it is known. How does this helper T cell get signal 2? Right? Remember that T cells and B cells need 2 signals to be activated. Antigen plus a co-stimulatory signal. And I don't think we know exactly why some people are stimulated by allergens. Most of us aren't because it's not harmful to the body. Yes?
Well, there is one theory that I heard that, surviving Neanderthal genes, within Homo sapiens, create a lot of the allergen effects. So they'll create some certain unnecessarily complements, or essentially create a bypass the systems that most people have.
So Neanderthals didn't have the hypersensitivity?
They did. That's the theory that I heard. Is that because of the Neanderthals had different genome, so a few of those genes survived, and then they're creating almost very similar, but also different mechanisms for these sort of things.
So, Alex, you're saying there's probably a genetic predisposition that's related to survival back in prehistoric times. Absolutely, I'm sure that there's a lot of research going on about that, especially given that more and more people are developing allergies. We do know that this response didn't evolve against allergens. It evolved to combat parasites, usually worms or larger extracellular pathogens. But in our modern society, and part of the hygiene hypothesis states that because we're not exposed to many parasites anymore, we live in a very hygienic society, we don't respond to the appropriate allergens or the appropriate triggers. The body kind of isn't taught what is an appropriate trigger and what is not an appropriate trigger because we have so little exposure early in life. That's the hygiene hypothesis. It's still being studied, but it's gaining a lot of support and a lot of evidence in the literature. So what happens that first time you're exposed, the allergen binds to the T helper cell. Somehow it activates and triggers the T helper cell to secrete cytokines. At the same time, you may have a B cell that also recognizes that particular allergen. Since your T helper cell was triggered, it can provide the co-stimulatory, it can help activate the B cell, and some of these cytokines will tell the B cell to actually make IgE antibodies. So interleukin-4 is a cytokine T cells produce. If the T cells produce it and there's B cells around, interleukin-4 tells the B cell to make IgE type of antibodies. So that's your first exposure. Now these IgE antibodies are specific to that antigen, and they're going to circulate through the body. And they're going to bind to any mast cells or eosinophils that happen to be around, because that Fc portion will bind there. So now, on your second exposure, you have these mast cells that are bound to IgE antibodies. And when that allergen comes in to the body, it'll trigger those IgEs, immediately causing the release of the granule contents, histamine, proteases, as well as prostaglandins and leukotrienes. So it's going to secrete. Histamine is the most important one. It's the one that we're most familiar with. That's why Benadryl, an antihistamine, is what you take if you have hives, because it's blocking this degranulation. It's blocking histamine's response. So the main mediators to know about are this histamine, because that's one we can affect. Prostaglandins and leukotrienes are made by the cyclooxygenase pathway. So membrane phospholipids are clipped and degraded into prostaglandins or leukotrienes. And the reason it's important to know these are that cyclooxygenase, which is an enzyme that creates prostaglandins, is the target of ibuprofen. It's also the target of Vioxx or Celebrex. Those specifically bind to COX-2, so they are more targeted for certain types of inflammation. So we use drugs all the time that are interfering with this pathway to try to calm down in this hypersensitivity response. For people with asthma, one of the best asthma drugs is a leukotriene inhibitor. So to try to prevent that bronchial spasm, you can block this pathway as well. So those are important mediators. And again, this happens immediately after exposure to that antigen, but only on the second exposure. I want to show you the timeline. So here's your exposure. Almost immediately, within 5 minutes, up to 30 minutes, you'll start to have this response. And then once those mast cells have released all of their granules, that response starts to go down. So most of the time when you have hives, you're really, really itchy, and it kind of goes all over the body for about an hour, and then it goes away. But you'll also likely have a lingering skin reaction, and that's the second late phase reaction. And what's causing it is other types of cytokines. So in addition to degranulation and producing prostaglandins and leukotrienes, those cells, those mast cells also are going to produce cytokines. In addition, the T cells, when this antigen comes in, are again activated, the T helper cells, to produce cytokines. So you have mast cells, T cells, T helper cells, and also eosinophils, which are producing cytokines, causing some local inflammation. So that's this late phase reaction. It's not late in the sense of what we talked about with acute versus chronic inflammation. This is actually a combined effect of acute and chronic inflammation. The reason is that you're going to stimulate macrophages and neutrophils, acute inflammatory mediators. Also, you've stimulated your helper T cells and your macrophages, so you also have a chronic. So you could call it acute on chronic or mixed inflammation. But it's late phase in that it's not that immediate degranulation that causes the initial response to an allergen. And quickly, I'm going to talk about what you'll see clinically, which most of you already kind of know. You can have allergies that have local effects, and then you can have systemic allergies. Oftentimes, how you respond depends on how that allergen gets into the body. A bee sting, for example, goes directly into the bloodstream and can cause anaphylaxis to somebody who's allergic. If you have a peanut allergy, it depends on how that antigen gets in. Some people with a peanut allergy can touch peanuts and be fine, but as soon as they ingest them, they're going to have massive diarrhea. Some people, if they inhale peanut dust, will get bronchospasm and will get an anaphylactic type response to it. So the type of reaction depends on how you get exposed to the antigen. So it can be local. It can be a combined local. You can have some respiratory and some skin manifestations, or you can have a systemic, and the one we're all familiar with is anaphylaxis. So local responses, if it's respiratory, that's when you usually get sinusitis, mucus secretion. In people with asthma, you get airway swelling and bronchoconstriction. In the GI, you get diarrhea and inflammation, so just general GI discomfort, pains, bloating, that type of thing from inflammation in the GI tract. And then in the skin, you'll usually see it in the form of a rash. Sometimes you'll get blisters. Systemic type 1 results in anaphylaxis. And that's where you get all of this at once. And the biggest problem with anaphylaxis is, of course, bronchoconstriction, where you can no longer breathe. The airway tightens, you get hypersecretion of mucus, you get bronchospasm of the smooth muscle of the bronchial airways, and you get edema or swelling of that tissue, which is what's showing here. So this is a normal larynx. And in somebody who's undergoing anaphylaxis, this is what it looks like. You can see all of this tissue here is swollen. That's from histamine and other of those vasoactive amines that immediately increase vascular permeability and cause fluid to release and dump into the tissue. So you get all of that edema there. Anybody know the treatment for anaphylaxis? Yes. An EpiPen. Anybody who has a severe allergy should be carrying an EpiPen. So why do you think epinephrine is a good treatment for anaphylaxis? Yeah? So it stops blood flow to the site. How would it do that? It does. It's a vasoconstrictor. This is kind of going back to some of your anatomy and physiology. Did you learn about the adrenergic system? The sympathetic and parasympathetic system, which are largely mediated. Your sympathetic system is driven by epinephrine and norepinephrine. Yes? So I'm not going to go over it in too much detail here. If you've forgotten about it, you can go back and review it. But just remember, your sympathetic nervous system, that's your fight or flight nervous system, is largely driven by epinephrine. And so vasoconstriction is mediated by alpha-1 adrenergic receptors. Don't do that. Are we still recording? Yes. Which are on blood vessels and cause vasoconstriction. Yes?
Don't the beta-2 receptors cause bronchodilation?
Yes. Now, you're a TA for anatomy and physiology, so very good. You also get bronchial relaxation, which is mediated by the beta-2 receptors. Awesome. So that's why we give epinephrine for anaphylaxis. We're trying to counter the response. At the same time, you should also give antihistamines. But most people, you just need the epinephrine to get through that acute phase, that immediate part, and then get some kind of antihistamines on board as quick as possible. Questions over here? Yes, Alex. Okay, Ian.
In systemic anaphylaxis, can you get shocked from the loss of blood pressure from all the fluids kind of shooting out?
Yes. So the question is, with anaphylaxis, can you get shock? And if you get swelling throughout the body, if enough mast cells are stimulated and you get vascular permeability throughout the whole body, fluid is going to leave the blood and go into the tissues of the body. And when that fluid leaves the blood, your blood pressure goes down. So shock is defined by a particular percent drop in blood pressure that causes end organ damage, and that can definitely occur with anaphylaxis. But most of the time, people are going to suffocate before they die from shock. Okay. That was it? Okay. Oh, you were just helping. Got it. So it's really important to remember your anatomy and physiology. It does come back into a lot of pathology. Okay. Are we ready for a break? Yes. 2 minutes. Immediate hypersensitivity is the most complex. We're going to kind of fly through the next three because really you already understand the mechanisms because we've already gone over it with the immune system. All that's happening different is it's responding to something inappropriately. So antibody-mediated hypersensitivity, the trigger could be anything. It could be self-antigens. It could be microbial antigens. And this actually happens a lot. Let's say you get an infection, and your body fights that infection. And some of those antigens that were on the microbes get into the blood, and they circulate. And they go and they get deposited in a different area. So now you have antigens that are not actually part of the microbe, and they don't need to be fought, they don't need to be destroyed, but because they're in the wrong spot and you have antibodies to them, you mount an inappropriate response to them. So an example of that, which we'll get to when we talk about kidney diseases, is post-streptococcal glomerulonephritis. Happens in kids actually quite a bit, where you have strep throat or some other streptococcus infection, and some of those microbial antigens get into the bloodstream. And they get filtered by your kidneys, because your kidneys are your main blood filtration organ. And they get into the glomeruli of the kidneys. So your antibodies recognize those streptococcal antigens in the glomeruli of your kidneys and start to attack your kidneys. So you have an inappropriate response there. So microbial antigens. Environmental antigens can also cause this. And the mechanism or the pathogenesis of what happens is simply what you already have learned antibodies can do. So if antibodies bind to free-floating cells, such as platelets or red blood cells, that's what you see over here, the antibodies are opsonizing those cells, telling your body to phagocytose them. So this thrombocytopenia purpura is a disease where you create antibodies against platelets, and so you reduce your body's platelets. So the antibodies bind to your own self-platelets, and when they go through the spleen is your blood filtration organ. You have a lot of lymphocytes there. They recognize the antibodies, and that triggers phagocytosis of your platelets. So they get destroyed in the spleen. The other thing antibodies can do, remember antibodies can activate complement. They can activate macrophages. Those macrophages will secrete cytokines and induce acute inflammation and or chronic inflammation. So here you have so what happens in post-streptococcal glomerulonephritis, antibodies binding to these streptococcal antigens in the glomeruli trigger complement, they trigger macrophages, they secrete cytokines that call into the kidneys other leukocytes. So you end up with a lot of neutrophils, a lot of macrophages, and a lot of other cytokines in the area. And people with this will undergo acute kidney failure because of the inflammation. Basically, the inflammation blocks up the normal function of the glomerulus, which is to filter blood, and that filtrate is going to become the urine. And if that system gets blocked up, then it doesn't work anymore. We'll talk about it in kidney diseases. Then the third thing that can happen in some unique cases is those antibodies can actually disrupt the function of the cell, of normal cells. For microbes, we want that. We want antibodies to, for example, if they bind to a spike protein on a virus, it blocks the ability of that virus to get into a cell. That's a good thing. But if these antibodies are binding to your own cells, and this is what happens in Graves' disease, they're binding to the TSH receptor on your thyroid cells. The TSH receptor becomes chronically activated, and you just secrete a ton of thyroid hormone, thinking that the body needs it. So this is a cellular dysfunction caused by the antibodies. They're actually causing the cells to do the wrong thing. So 3 different ways that antibodies can mediate immune reaction. Here's a picture of the kidneys in Goodpasture syndrome. So Goodpasture syndrome is another kidney disease where you generate antibodies that bind to the basement membrane in the glomerulus. It also binds to basement membrane proteins in the lungs. So people with Goodpasture syndrome have a lot of antibodies that are binding to their glomeruli. So the green here is an immunofluorescence, a dye stained for IgG. So it'll bind to any IgG. And you can see the IgG is just coating all through the glomerulus here. It shouldn't be there. This over here is a renal tubule. This is background. So the bright parts over here are where you see IgG. Questions about antibody-mediated? So immune complex is very similar. The triggers can be, again, self, microbial, or environmental. And what happens is the antigen is free. Rather than being part of the tissue, it's free floating in the blood. And so it complexes with the antibodies. So you actually, here's a good little drawing of that. So you get antibodies that are binding and forming these big complexes. Not the antibody's fault, it's just doing what it's supposed to do. These immune complexes then will deposit, usually in the blood vessels, because they're circulating in the blood, so they just get trapped in the wall of the blood vessels. Or in the kidneys, so the glomeruli, because they're constantly filtering the blood, the glomeruli are particularly susceptible. And then sometimes you'll see it in the skin. But the blood vessels, vasculitis and glomerulonephritis are the two main clinical manifestations of this. And then same thing, these antibodies do what they're supposed to do. They trigger complement, they trigger macrophages, and they create an inflammatory response. So here's an example of vasculitis. Vasculitis is often associated with immune complexes. And you've seen this picture before. So take a minute to think about which arrow matches up with which finding on this histology image. You can talk to each other.
I gotta go a little early.
That's fine. Thanks.
We'll have materials for hypersensitivity for Friday of next week.
Sounds good.
That's what you guys said. You're thinking that.
Which one is this pointing to? This arrow right here. What is it pointing to? There are lots of it. So it's actually meant to be pointing to inflammation. Although it could be pointing pretty much anywhere in this slide, because acute inflammation is going on everywhere. So all of these little purple dots are inflammatory cells. It's not high enough power for you to be able to tell what kind of cell they are. But there's a lot of cells in this area. So the reason that it's pointing right here is that this is roughly the outer wall of the blood vessel, and this is roughly the inner wall of the blood vessel. And you can see all of these inflammatory cells in the wall of the blood vessel, so they shouldn't be there. It's not where you would normally expect to see cells. What about this right here in the middle? Inside the lumen of the blood vessel, what do you find? Microthrombi, yes. So these are the We've not gone over this before, so it's fine if you didn't get it. This is really just meant to get you thinking and looking and trying to observe what's going on here. So in thrombi, and we didn't have a chance to talk about it before, but thrombi form in blood vessels. Thrombi are blood clots. So they're going to form where you have your coagulation factors and platelets. So vasculitis, typically you get a lot of inflammatory cells in the wall of the blood vessel, and those inflammatory cells are producing cytokines that activate the coagulation pathway and activate platelets to form little blood clots inside the blood vessel. Yes? Good question. So what are these? That is a histology processing error. It's very hard to get perfect slides because you want to really thin slice the tissue. So this is just where the tissue kind of tore, or there's a bubble under the slide. So it's not part of the actual image. And then remember over here, so by process of elimination, we have fibrinoid necrosis. So remember, fibrinous inflammation is caused by fibrin from the blood leaking into an area. Fibrinous necrosis is necrosis associated with fibrinous inflammation. It's when you have a lot of fibrin leaking in because of vascular permeability, because in this case, those immune complexes deposit into the wall of the blood vessel. They trigger inflammation. They trigger edema, vascular permeability. And because protein is leaking from the lumen into the wall of the blood vessel, it's causing fibrinoid necrosis over here. One last image. Oh, yes.
Do microthrombi, can they like bond together to form like a
Yes. Question is, can microthrombi form big thrombi? And yes, they can, and it's a very big risk. So oftentimes you'll get clots, big clots with vasculitis. Okay, just this last image is just showing you immune complexes in the skin in a patient with lupus. So what we're staining for here is C3, complement 3. So you can see these antibodies have triggered complement. These antibody immune complexes deposit in the skin and they trigger complement, and so the skin is full of inflammation. All right, have a great weekend.